A very big hello to all my wonderful PU1 champions. I hope all my dear children are doing great. So please listen to me very carefully. This is a passing package. There will be a list of questions given to you from all the 19 chapters. But since there are 19 chapters, obviously it is not very much easy to cover up all the questions individually. So I'll be giving you the list of questions towards the end. Initially in some chapters, I will definitely go ahead and explain you the answers as well. But yes, as I said, towards the end, you would be seeing that I'll be showing you just the list. So I wanted to make that clear and very transparent with you guys. And also, if you are someone who is having no idea as in where to begin from, then probably these questions could help you, and that's the only reason we are putting up this particular video. There is no blueprint that is given. Obviously your schools or PU colleges have the right to give the questions according to their own will, since there is no common blueprint that is being followed. I hope you have an understanding of that. If at all your teacher has already told you that these questions are super important and things like that, focus on them as well. Okay, I hope it's clear. And if you feel that this video is helpful for you, definitely let us know in the comment section. Wishing you all the best, my dear children. You are going to do good. Just give it a try. Okay, starting off with the first thing that we have, the cell, the unit of life. As I said, this is according to the previous blueprint which was there. According to that, it was 12 marks. This chapter holded 12 marks. And there are certain 2 markers that usually came in. First one is state cell theory. State cell theory, in that you just have to say that 2 things. First thing, that all living organisms are composed of what, my dear children? Cells, and also the product of cells. This was given by Schleiden as well as Schwann. Later on, Virchow came into the picture and he said that cells which you see today are not just coming from air. Basically, they come from the pre-existing cell. So probably you had a cell before, and this cell, my dear children, later divided and now you have a cell. So this cell came from what? The pre-existing cell. This was given by Virchow. Before that, Schleiden and Schwann gave that all living organisms are made up of cells and also products of cells. That is cell theory. Now what are mesosomes? Mention any one of the function. Mesosomes, basically, my dear children, it is found in the case of prokaryotes, not in the case of eukaryotes. Always remember. And they are extensions of what? The plasma membrane. So basically, suppose your plasma membrane is something like this, forming something like this. This structure which you have is known as now mesosome, my dear children. And it has very important functions, and also it comes in different forms. Sometimes it can be like this, such as vesicles, or it can form tubules, or probably it can be in the form of lamellae as well. And what is the use of this? Basically, there are some components stored inside the mesosomes which are going to help in cell wall formation, DNA replication, as well as distribution to the daughter cells. Now listen to me very carefully. In the case of eukaryotes, that is advanced organisms, my dear children, or advanced cell in that particular case, they have nucleus, they have multiple organelles to do all this function. But prokaryotes are not like that. Prokaryotes are very much primitive. They do not have membrane-bound cell organelles. They do not have a nucleus also. So basically, the functions which have to be performed really well are segregated to different parts. And one such part is mesosome. So here it is the one that helps in cell wall formation, DNA replication, and also after replicating, it also makes sure that it passes down to the daughter cells as well. Now they also help in respiration. For the process of respiration, you need what? A lot of enzymes. You have studied that, right? Now these enzymes are spread throughout the plasma membrane, or basically the membrane of the mesosome right there, which helps in what? Respiration, and also in the secretion process to increase the surface area of the plasma membrane and enzymatic content. So my dear children, basically this mesosome which is there, when it is like this, you are increasing the surface area for the enzyme to be attached and due to the process of what? Respiration as well as secretion, all those things. So mesosomes are found in what? Prokaryotes. I hope that is pretty much clear, and they are extensions of plasma membrane as well. Now the next question, list the types of ribosomes found in prokaryotes and eukaryotes. Now in prokaryotes, my dear children, so starting with eukaryotes, eukaryotes have ribosomes with ATS. 8S stands for Swedberg's unit. Swedberg's unit is basically a calculation for the sedimentation coefficient. The rate of sedimentation is being understood with the help of this particular unit out there, and it is ATS for eukaryotes. Now for prokaryotes, my dear children, it is going to be lesser than that. What is it going to be? It is going to be 70S. It is going to be 70S. Now if at all you know, ribosome which is there, it is made up of 2 subunits, one large subunit and one small subunit. Now if at all I talk about the large subunit separately and the smaller subunit separately, both of them will have different rate of sedimentation. If you talk about the ATS ribosome of eukaryotes, the larger subunit, the sedimentation rate of it comes out to be what? It comes out to be, do you know what is exactly it is? It is 60 as well as So 60 is the larger subunit and the smaller subunit, it is 40. So 60 plus 40 is equals to 100. No, man, then how does 80 come? Listen, if at all you take the ribosome as a whole, the rate of sedimentation of ribosome as a whole is 80s. But if at all you take the larger subunit, it is just 60s. If at all you take the individual smaller subunit, it will be just 40s. That is why you have 60s and 40s right over here. Please remember that. Similarly, in the case of the prokaryotes, when I say it is 70s, the larger subunit is going to be 50 and the smaller subunit is going to be basically 30s. So 50 and 30. 50 and 30. Got it? 50 and 30. So 50 and 30 is equals to 80. Should it be like that? No, no, no. So you remember like that. Okay. 80s will not be broken down to 50 and 30. 50 and 30 is for 70 only. And 60 and 40 is for 80. Please do not forget it. Okay. S stands for Swedberg's unit. Now, next thing, difference between RER and SER. RER is what? Rough endoplasmic reticulum. Why is it rough? Because this endoplasmic reticulum, on its surface, my dear children, there are ribosomes, which makes its appearance rough under the microscope. And smooth endoplasmic reticulum is basically is not having what? The ribosomes, which is why it has a very smooth surface. So what all do you need to say? Rough endoplasmic reticulum has ribosome on its surface, making it appear rough, while smooth endoplasmic reticulum lacks ribosome and has a smooth appearance. Now you have to talk about the function also. My dear children, whenever you have the term ribosome, you know that ribosomes are super important for the process of protein formation. If at all you didn't know that right now. Okay, they are super important for the process of protein formation. So wherever ribosomes are there, protein formation will definitely happen there. So RER, since it has ribosome, Baba, it will be helpful in what? Protein synthesis as well as processing. Whereas when you talk about SER, it is responsible for some other things synthesis. For example, lipid. For example, detoxification of things, and also for calcium storage. So what all things are there for SER? SER is lipid synthesis, as well as you have what? Detoxification being done by SER, and also it is super important for calcium storage. You know, in our muscles, we need a lot of calcium. And in our muscles, you have a modified version of SER, which you call as sarcoplasmic reticulum, which is responsible for the calcium storage itself. So please remember, RER is for protein synthesis, whereas SER is for what? SER is for lipid synthesis, detoxification, as well as calcium storage. I hope that is clear. Yes? Okay, by the way, my dear children, if at all you think that my pace is a bit fast, you can just slow down the video in the settings section, because yes, as I said, there are so many chapters, which is why, and you have less time, which is exactly why I am just speeding it up. And also one more very important thing, when I show you the list of the questions, and if you feel that I needed an answer for this question as well, you can put down the question in the comment section. I'll try my best to answer at the earliest. Okay? Okay, fine. Now name the 4 endomembrane organelles. Endomembrane organelles, my dear children, they are related. They work parallelly, as in one product comes and then the organelle will be acting on it and then it goes sequentially. So basically the endomembrane system includes the endoplasmic reticulum. Now whatever endoplasmic reticulum is making, someone is going to modify it and package it, and that is Golgi apparatus. And further, my dear children, from Golgi apparatus, you are going to get different type of lysosomes and also vacuoles. You get it? So basically endoplasmic reticulum, Golgi apparatus, lysosome and vacuoles, they are known as the part of the endomembrane organelles, and because their functions are all what? Coordinated together. That. Now functions of Golgi apparatus, SER, lysosome, vacuole, chloroplast and mitochondria. These things are being asked. So Golgi apparatus, as I told you, it is responsible for modification. The proteins that came, the lipids that came from the endoplasmic reticulum, they are going to modify it up over here. They are going to be sorted and they are going to be packaged. The proteins and lipids are going to be packaged for what? The transport process. Now over here you have smooth endoplasmic reticulum. We already discussed about it. It is responsible for lipids, detoxification of harmful products, and that's all. Now the next thing that we have is lysosome. What is the use of lysosome? Lysosome is an organelle, my dear children, where you would be having so many enzymes stored in it. It is digestive enzymes. Basically, suppose the cell which is there, inside the cell a foreign particle has come through. Now you want to kill the foreign particle. What does the lysosome do? Lysosome will be releasing the enzymes which might be killing the foreign particle and also sometimes the whole cell as well. Suppose a cell is in danger, it can affect the other cells also. It will act like a suicide bag and kill the whole cell by releasing the enzymes that it is storing. So basically I can say it breaks down the waste materials and cellular debris using the digestive enzyme that it has. Now vacuole, my dear children, you might have seen that the plant cell, in that 90% of the space is occupied by this big vacuole. Vacuole is responsible for what? It is responsible for storing nutrients, waste, and also, nutrients, water, and also some very not so important waste as well. That is the use of vacuole. Apart from that, vacuole is also responsible for providing structure to the plant cell. Okay, in our case also we have vacuole, that is an animal cell also you have vacuoles, but it will be very small. Now chloroplast, my dear children, it's a type of plastid which is responsible for doing photosynthesis. Why? Because it has chlorophyll pigments. Okay, mitochondria, what does it do? You all know it is responsible for production of ATP, adenosine triphosphate, very very important. And basically mitochondria is a site where aerobic respiration is going to happen, where the glucose finally is going to get broken down to CO2, H2O, as well as lots of ATP. So I'll say it's a place where ATP is going to be formed. Okay, so yes. Now what is active transport? Give an example. Active transport basically means that if at all I want to transport something out from inside, or basically in or out, whatever it is, and for that transport you are ready to spend some energy. You are ready to spend some energy. This is known as active transport. So transport in which ATP is utilized and is called as active transport. For example, you might have seen in sodium potassium channels and etc. like that, or sodium potassium pumps as well. So wherever you are making sure that the ATP is being spent or is being utilized, you say that this is involved. This is involving an active transport up there. Done? Done. Now we have 3 marker questions which usually comes from this chapter. List out the functions of plasma membrane. What is the function of plasma membrane, Abba? It is definitely, first of all, it is going to regulate what is going to enter and what is going to exit. So that is the one very important thing, selective permeability. Selectivity is saying that you can get in, you can get out. Okay, second thing, provides mechanical support and maintains the cell shape. So for that we need what? The plasma membrane. Next thing, facilitates cell communication through receptors. So my dear children, on the membrane you have so many receptors, and onto the receptors some proteins can be binding, which gives a signal as to basically what will happen inside the cell. Is there some secretion that is needed to happen? All the cell communication happens through the receptors which are there on the cell membrane, and that is why we say that it is helping in cell signaling and transport, okay, via the protein. So these proteins which are there, they are going to help in the cell signaling as well. Anyway, plasma membrane is involved here. Yes, yes. So receptors and all the proteins are super important for cell signaling or cell communication to happen. Fine. Now classify the types of plastid based on the type of pigments. Okay, so we have 3 types of plastid. One is chloroplast, which has the green pigments, that is chlorophyll, my dear children. It is responsible for photosynthesis. Now we have chromoplast, as in colored plastids out there, and over here it contains carotenoids, that is the what do you call, orange and the brown pigments out there. Not orange pigments, orange and the red pigments out there. Apart from that, you have xanthopils, which are the yellow pigments. All of them, my dear children, you see that there are different colors being formed, right? And you see different colors in which part of the plant? Generally in the flowers as well as fruit. So mostly you find them in the flowers and the fruits. Chromoplast are there, but apart from that it's there in the leaf also sometimes. Okay, now talking about leucoplast. Leucoplasts are the ones which are colorless in nature. They are plastids which are colorless in nature. They are responsible for storing starch. If they are responsible for storing starch, you call them amyloplast. Sorry, if on the other hand they are storing proteins, you call them aleuroplast. And if at all they are storing fat, you call them what? Elioplast. Okay, so first of all, amyloplast, that stores starch, that is carbohydrate, and proteins, then you call this as aleuroplast, and if at all it is storing fat, you call this as elioplast. So elioplast, I always remember it has some like oil term, and oil is basically fat. So you can also remember like that if at all needed. Now cell wall is non-living rigid structure made of 3 layers. Mention the wall layers and its chemical components of the bacteria. I'm talking about bacteria and I'm saying that it is basically having 3 wall layers. What are the 3 wall layers in the case of bacteria? First one that we have is glycocalyx. It can be in the form of capsule, which is going to be thick, or it can be a slime layer which is made up of polysaccharide. Either way. So glycocalyx is the first thing that you have. Second thing that you have is cell wall. Cell wall in the case of bacteria is made up of peptidoglycan itself, and this provides rigidity to the cell. Now plasma membrane, my dear children, is the next thing that we have, the innermost thing that we have, and it is made up of phospholipid bilayer. We know that. So glycocalyx, it can be capsule or slime layer, but remember it is made up of polysaccharide only. Cell wall, it is made up of peptidoglycan, and plasma membrane, my dear children, it is phospholipid bilayer. Then this is the case of bacteria. Now name the 3 components of bacterial flagellum. So flagellum, my dear children, looks like this. Over here you have a filament which moves in the which helps in the movement. That is the first thing, long helical structure for movement. Next thing that you have is hook, connects the filament to the basal body. So this is the basal body, and over here you see this particular part which is lightly shaded, this is known as hook. So that is responsible for connecting the filament to the basal body. Now basal body is what anchors the flagellum to the cell membrane. So it is the one that connects the whole flagellum over here to the cell membrane out there. So if at all I zoom this and I show it to you, it looks something like this. It looks something like this. So this is your filament, this is your hook, and this is going to be the basal body, which is going to allow the flagellum to be connected to the cell membrane. Okay, done. Now, what is cytoskeleton? And write any two functions of cytoskeleton and it's okay. So cytoskeleton is what? The network of the protein filaments which are there, which are going to provide the structure to the whole cell. Inside the cell, you need something to provide the structure, like just like how we have bones inside our body, similarly inside the cell they have cytoskeleton. And the functions of it, first of all, it maintains the cell shape and provides mechanical support. Apart from that, my dear children, it also helps in intracellular transport. If at all you want to transport something from here within the cell to somewhere else, then also you would be needing the help of cytoskeleton as well. Got it? Got it. Fine. Now, which organelle is called packaging apparatus and why? You know, I told you Golgi apparatus is the one that is responsible for packaging, and the reason, because it modifies, sorts and packages proteins, not just proteins but also lipids and anything that is formed by the endoplasmic reticulum. Got it? Yes. Next thing, difference between prokaryotes and eukaryotes. Prokaryotes are primitive, P for primitive and P for prokaryotes. And eukaryotes, my dear children, they are modified organisms out there. In the case of eukaryotes, as I said, there will be a nucleus. Inside the nucleus, my dear children, we have the DNA and everything. But in the case of prokaryotes, it's not like that. They just have a normal region out there. They just have a normal region. So, just a second. They have a normal region where the genetic material is just lying up there. So basically this region is known as nucleoid. So prokaryotes do not have a true nucleus. The genetic material is present in the nucleoid region. Whereas eukaryotes have a well-defined membrane-bound nucleus. That is the first thing. Now organelle also not found. The membrane-bound organelles are not found in the case of prokaryotes. They have ribosomes, but ribosomes is not membrane-bound. So over here, the prokaryote cell lack membrane-bound organelles such as mitochondria, Golgi apparatus and endoplasmic reticulum, while eukaryotes contains all these organelles out there. Now cell size, if you talk about prokaryotes, they have two cells, my dear, and bacteria, they are prokaryotes, right? So do you see bacteria out there? No, with our naked eyes we cannot see that. So prokaryote cells are generally smaller, 1 to 10 nanometer, compared to eukaryotic cell which are larger, 10 to 100 nanometer. So 1 to 10 nanometer is a normal size, you just think. And then this is what? 10 to 100. Fine. Now DNA structure, in the case of prokaryotes, DNA is what, my dear children? Usually the DNA which is up there is going to be circular in nature. Whereas in our case, the DNA, that is eukaryotes, my dear children, DNA is going to be linear. And also remember one very important thing. In our case, the DNA has to be put inside the nucleus, which is why it has to make it has to be made very, very compact. And to make it compact, we need help of some proteins which you call as what? Histone proteins, which you call as histone proteins. And over here, you do not need to fit in anywhere, which is why you do not need so much amount of compaction, and also their DNA is not too long also. Okay, so which is why they do not have histone proteins. So these are a few things that you need to remember between the prokaryotes as well as eukaryotes. Now what do you have? You have the 5-marker questions. These are the usual question that comes in. There are diagram based questions. Go through each of them. Okay. So I'm not going ahead in depth. You can clearly see that. Please take a screenshot of this and go ahead, start solving these questions. What you can do is, if at all you open the chapter, put Control F on the keyboard and write it down, the term, whatever you're searching for, whatever paragraph comes, read through it. If at all you do not have much time left, I'm asking, if at all you do not have much time left. If you have time left, go through the chapter, at least watch one short video, that will be more than enough. Understand the concept. Okay? Now name the following powerhouse of the cell. You know, Baba, it is what? The mitochondria. Why powerhouse? Because it is producing ATP, site of steroidal hormone synthesis. Steroidal hormone synthesis is basically, it is going to be smooth endoplasmic reticulum. Now reservoir of hydrolytic enzymes, Baba, hydrolytic enzymes are basically digestive enzymes only, that is lysosomes. Now packaging units of the cell, it is Golgi apparatus. Locomotor structures of the cell, it can be cilia, it can be flagella also. So rest of the things are given, diagram based questions are given. Look at the diagram, make sure you draw them, practice them, that is super important. And over here also one more question was there, diagram of mitochondria and also chloroplast, very, very important. Okay, now animal kingdom, my dear children, I have made a one-shot video on it. If at all you want to watch it, you can watch it if you have time. If not, listen to me carefully. Okay, so you might have done your studies. See these questions as well, but do other questions also. Now, what is bioluminescence? Give an example. Bioluminescence is the ability of an organism to produce and emit light. You might have seen it, right? What do you call those flies as? I forgot. Fireflies. Okay, fireflies, they show bioluminescence are there. Similarly, so fireflies, certain jellyfish and some deep sea fishes like anglerfish, my dear children, all of them are examples of the organisms that show bioluminescence. Now, all vertebrates are chordates, but all chordates are not vertebrates. Justify. Listen, this is very, very simple. Always remember, chordates that you have over here, my dear children, they have notochord. What do they have? They have notochord. Now, there are three categories of organisms. One is Urochordata, one is Cephalochordata, and one is Vertebrata. So what exactly happens is that here in the case of Urochordata, notochord is there, but in the tail region only, and that too in the larval stage. Notochord is there, but only in the tail region, and that too in the larval stage. Cephalochordata, head to tail, it is there, notochord is there, and it is there throughout the lifetime. In the case of Vertebrata, my dear children, this notochord which is there, it is now converted into something else that is known as vertebral column. So they do not have notochord anymore. Instead, they would be having vertebral column. So can I say that all vertebrates are chordates? All vertebrates are chordates, but can I say that all chordates are vertebrates? No. All chordates are not vertebrates, but all vertebrates are definitely chordates because they come under chordata. That is the reason. So chordates have a notochord at some stage of life, but not all develop a vertebral column. Vertebrates are a subgroup of chordates where the notochord is replaced by the vertebral column. Example, Amphioxus, which is lancelet, is a chordate but not a vertebrate. So lancelet which is there, or Amphioxus which is there, it is a chordate but it is not coming under vertebrate. Just to give an example if at all you want. Now difference, not if at all you want, it will be really good if at all you do. Okay. Difference between coelomates and acoelomates. What do you mean by that? Listen, in our wall layers, we have ectoderm, endoderm and mesoderm. Ectoderm is the outermost layer, endoderm is the innermost one, and in the middle of both these things, we have mesoderm. Suppose you have a cavity over here, and on both the sides of the cavity, you have the layer of mesoderm. You call this cavity as a coelom. What do you call this? Coelom. And organisms that has this cavity, they are known as coelomates. And most of them, they have it, okay? So animals with a true coelom, a coelom like this, my dear children, where both the sides are covered by the mesoderm layer, is basically, so animals with a true coelom, body cavity is lined by mesoderm. Example, annelids, mollusca, as well as chordates. They come under coelomates. Now there are multiple other examples also, you can remember at least a few of them. Now acoelomates means basically they do not have any cavity at all. And the example for that is basically platyhelminthes. So animals without a coelom, body cavity is absent, and the example for that is platyhelminthes. Now you have one more group, that is pseudocoelomate, where you have a false coelom. Yes, you have a cavity, but it is not completely lined by mesoderm, which is why you call them pseudocoelomates, and that is usually found in aschelminthes. So general thing. Okay, this is a thing that you need to know. Next thing, differentiate between radial and bilateral symmetry. My dear children, radial symmetry is basically having a ball. This ball you can cut through any plane which passes through center. It has to pass through center, but you can cut like this, you can cut like this, you can cut like this. It is going to give you equal halves anyhow. This is known as radial symmetry. Body parts are arranged around a central axis, hence can be put into equal halves through any multiple planes that passes through the center. Examples of this is cnidarians and ctenophorans. Okay, cnidarians and ctenophorans. For example, in cnidaria comes jellyfish and hydra. Jellyfish, just imagine a picture, jellyfish. Okay, it's like this and it has lots of tentacles around. Okay, when you cut like this, you can cut like this, you can cut in any way. Similarly, hydra also. Both of them, they come under cnidarians. Apart from that, ctenophora are also like this only. Now bilateral symmetry, here the body can be divided only into two equal halves and only through one particular plane. For example, us, we can be divided into two equal halves just like this. Can we be divided like this into equal halves? No, that is not possible. So basically, body can be divided into equal halves only through one plane, found in chordates, found in arthropods, and found in annelids. Just think of things, okay, just think. Mosquito, that is coming under arthropoda. Earthworm coming under annelida. All of them you can divide only through one particular plane for giving equal halves. So that is the thing. Now, what is metamerism? Name an organism exhibiting this particular character. Metamerism means body is segmented externally and also internally in a serial repetition of parts. So there is a repetition of parts, okay, and there is segmentation also. That is known as metamerism. Example, you see that in earthworm, that comes under annelida. Now, difference between external and internal fertilization. Suppose, my region, the egg and the sperm, or basically the gametes of using outside the body of the female, it is known as external fertilization. For example, in frogs and fishes, they lay the eggs, everything happens, and onto the eggs, the sperm is being put. Remember, it happens outside the body, not inside the body. Whereas internal fertilization means fertilization occurring inside the female body. For example, mammals, in the case of birds, in the case of reptiles, all of them show internal fertilization. Okay. Fine. Remember over here, echinodermata show two types of symmetry. The adult form show different and larval form show different. If at all you remember it, put it down in the comment section right now. And that is an important thing, go search about it. Now name the animals exhibiting the given characters. Organism with a sucking and a circular mouth. Sucking and a circular mouth. Remember, vertebrates which are there, they are divided into two things. Agnathans, who do not have a jaw. Okay, they might have teeth and everything out there, they do not have a jaw to fit that in. Okay, and gnathostomata, which have the jaw. So agnatha have which is there, inside that this organism come, that is lamprey. Okay, lamprey, it will be something like this. The jaw is going to be, and it has circular mouth. Okay, it has a circular mouth and it is sucking mouth. So basically lamprey, they come under Ignatha cyclostomata specifically. Okay, and organism in which notochord is present only in the larval stage, I told you it's Eukaryote. Example you can give Ascidia. Example you can give Ascidia. So here all the organisms name are important for you to remember. The phylum is important for you to remember. So yes, it takes some time, but my dear children, we can do it. Now, difference between direct and indirect development. Direct development means young ones, my dear children. For example, in the case of human beings, your mother gave birth to you. It's just direct. Is there any stage, larval stage and any other pupal stage and things like that, where you have the tail and then you become like this? Once you are produced, you're just like a human baby, right? So basically here, the young one resembles the adult form without a larval stage. That is known as direct development. Whereas indirect development means the young one passes through larval stages before becoming an adult. For example, the frogs and the butterflies, they have multiple stages in between. That is meaning of indirect development. Now I mentioned the two functions of the flame cells. Flame cells, my dear children, they are super important. They are found in the case of flatworms, you call them platyhelminthes, and it is super important for the process of excretion. So flame cells helps in the removal of nitrogenous waste such as ammonia. What is the nitrogen waste? Ammonia. And osmoregulation, they maintain the water and ionic balance in the freshwater flatworms such as planaria. So flame cells is an excretory mode of excretion, is a one that helps in excretion in the case of platyhelminthes, and the nitrogenous waste that they remove is basically ammonia. They are also responsible for maintaining the water and ionic balance. Cool? Yeah. Example for platyhelminthes, planaria. Fine. Give any two parasitic adaptation of flatworms. Flatworms, as I told, flatworms, as I said, they are known as platyhelminthes. And the most famous example that we have is tapeworm. Tapeworm are found in our intestine, my dear children. And what are the adaptation that they should be having? Hooks and suckers, because they do not have hands and legs to hold on to like this. Okay, if at all they have, they would be stuck somewhere, which is why, my dear children, they have hooks and suckers. They're found in the tapeworms for firm attachment to the host intestine, and also high reproductive capacity. If one organism comes inside the body, they are going to make sure that they make a colony of them such that the infection goes on. So they produce a large number of eggs, ensuring survival despite host defense. So host obviously will be showing some defense mechanism to act against it. They would be having high reproductive capacity. Okay. Now, these are the other questions that you have in 5 markers. List 2 features of chordates, list 2 classes of chordates which are oviparous, and development is direct and characteristic of mammals. All of the questions, my dear children, you can clearly see over here. I gave the answer for this particular question also. Assign the organs of. So please note the functions of these organs as well. Very important. Okay. So these are the questions. If at all you want, you can take a screenshot. Cool. Next thing, plant growth and development. So ABA is also known as stress hormone. Give reasons. ABA is basically abscisic acid. And why do you call it as stress hormone? Because, my dear children, whenever the plant feels it is under stress. For example, it is grown in a place where there is too much amount of salt in the soil. Obviously, a plant cannot go at this particular point of time. ABA is going to be released. Okay, forget that. Suppose it is growing in an area where there is no too much amount of water. So obviously, it has to make sure that the water is not lost. Whatever water it has, it should not be lost. Which is why we should be closing up the stomatal pore. Who gives the signal for closing up the stomatal pore? ABA. So ABA is basically a stress hormone because it helps the plant survive during drought, during salinity, that is when extreme salt condition is there, during extreme temperatures, by closing stomata and inducing dormancy. It does not allow the seed to grow or germinate until and unless all things are favorable. So this is a stress hormone. Differentiate between dedifferentiation and redifferentiation. Okay, listen. First of all is differentiation. Differentiation basically means that the cell, suppose I am the cell, I have gained maturity, I am not going to divide anymore, I am just going to do the function that is assigned to me. This is known as differentiation. Now at one point I think that, oh, I should be switching to some other career, and again, so basically I am going to move into this phase that is known as dedifferentiation. I again have the ability to do divide now. I have the ability to divide now. And suppose one day I feel, no, I should be having a proper function. I go back. That is now known as redifferentiation. You get it? Where it has lost the ability to divide again. It has a proper function but has lost the ability to divide. Whenever you have the term differentiation or redifferentiation, just understand it has lost the ability to divide. So dedifferentiation is different, okay? Dedifferentiation, mature cells regain the ability to divide. So it was mature once and now it is regaining the ability to divide. Example you will see in the formation of cambium from parenchyma. So from parenchyma, when you form the cambium cells, the cambium cells has an ability to divide. They divide very, very rapidly. So that is dedifferentiation. Redifferentiation means dedifferentiated cells which were dividing now, they has lost the ability and they become again the specialized or the mature cells. So for example, formation of secondary xylem and the phloem from the cambium. From the cambium again secondary xylem and phloem is being formed. So that is known as redifferentiation. Now define plasticity, give an example. So plasticity is basically plant follow different pathways in response to environment or phases of life to form different kinds of structure. This feature is known as what? Plasticity. Basically the ability to change very much. So example, in cotton the leaves of the juvenile plant, the small plants out there, are different in shape than from the leaves of the mature plant. So basically always remember, in cotton the leaves of the small plants which are there, they are going to be different from the leaves of the mature plants which would be there. That is something that you see. Okay, it's because they are showing plasticity. The environments are going to change when they are going to grow. That is the reason that happens. Okay, now heterophyly is an example of plasticity. Justify. Heterophyly refers to different leaf forms in the same plant due to genetic, environmental or developmental factors. Listen, a same plant has different types of leaves. Okay, different types of leaves. Why? Because probably or because of the environmental conditions or some genetic factors or some development factors. Example, you see this in buttercup. Difference in shapes of leaf produced in air and those produced in water. So over here there is a picture. This is the one that lives in air, that is terrestrial habitat. This is the one that lives in water. Over here you see the structure of the leaf is entirely different. Why? Because they have to be better adapted to that particular place. Why it happened? But because of some genetic, environmental or some developmental factors out there. Got it? Yes. That is heterophyly. Buttercup is the example. Remember that. Now, what are plant growth regulators? Mention the types with the examples. Now, this is very frequently asked. Plant growth regulators as in the one that is going to help in the plant's growth. It can show inhibiting features. It can be inhibitors. It does not allow the plant growth, or it can promote also. So, there are two things: growth promoters and growth inhibitors. Growth promoters are three of them: auxin, gibberellin, as well as cytokinins. Growth inhibitors are only two: abscisic acid and ethylene. Remember, ethylene is the gaseous hormone. It is what? A gaseous hormone. Now give an example each for natural and synthetic auxins. Listen, natural auxin is indole 3-acetic acid. You call this as IAA. Synthetic auxin is 2,4-dichlorophenoxyacetic acid. You also call this as 2,4-D. Apart from that, also you have multiple examples. Listen to me. Auxins like IAA and indole butyric acid, IBA. So IAA and IBA, both of them are natural auxins. Whereas on the other hand, you have naphthalene acetic acid and 2,4-D. That is the same thing that we discussed over here. Both of them, my dear children, are synthetic auxins. They have been chemically made. They are not naturally obtained. You not naturally obtain it from any plants out there. Okay, so please know their names also. So name the gaseous PGR, that is plant growth regulator, and mention its application. Gaseous PGR that I know is definitely ethylene, which is a growth inhibitor. And also, my dear children, always remember they are responsible for fruit ripening. They are responsible for what? Fruit ripening. So you might have seen that banana, one ripe banana, if you keep between any raw banana out there, my dear children, they also become ripe. Why? Because these gaseous hormones are being released out there. Okay, and also they are responsible for flower induction in the case of pineapples. In the case of pineapple. Okay. Now over here, what is respiratory climacteric, and name the growth hormone responsible for it. So it is a sharp rise in respiration rate during fruit ripening. Listen, respiration rate. Why do you need to increase the respiration rate? Because probably you need to produce too much amount of ATP, because the end result of respiration is what? ATP. Now why do you need the ATP during fruit ripening? Because, my dear children, during fruit ripening there are so many enzymes which are working. Amylases which will be acting on basically the carbohydrates up there, breaking it down. Pectinases which will be acting on the pectin, that is a part of the cell wall. Cellulases which is going to break the cellulose. All of this, my dear children. So why are we doing that? So breaking down the starch to soften the cell wall and convert the complex sugars to simpler ones. for which you need energy. So when all these enzymes are working for the formation, for basically the ripening of the fruit, you obviously need some energy to be put in for these enzymes to work, right? So which is why we say that there is an increase in the respiration rate. And who is responsible for this process? Which PGR is responsible for this process? It is basically ethylene. It is basically ethylene. Always remember that. So enzymes like amylase, which breaks down the complex sugars to simpler sugars, pectinase, which is responsible for softening of the cell wall, even similarly cellulase also for softening of the cell wall. So at this particular point of time, you need high amount of energy. Now graphically represent the ideal sigmoid growth curve. Sigmoid growth curve. Sigmoid means S-shaped curve. Okay, it is what? S-shaped curve. S-shaped curve is going to be obtained whenever there is a stagnancy in growth. So it is going to grow. Whatever organisms are there, whatever thing is there, it is going to grow, but only up to a limit. So that is why you get a sigmoid curve. Now over here, you are being asked to graphically represent it. So make sure you write it properly. This is from your NCERT itself. So time has to be mentioned over here, and size, weight of the organ has to be mentioned over here. The lag phase is there. Then there is exponential phase where it is going to grow, and then there is a stationary phase beyond which it is not going to grow. It will be stationary only, constant itself. Okay, so that is sigmoid curve that you get. Apart from that, you have J-shaped curve also. Please know about that also. Cool. Now briefly explain, this is a 3-marker question. Briefly explain the physiological effects of auxin, gibberellin, cytokinin, and ethylene, and abscisic acid. Auxin basically is responsible for cell elongation. If at all your cell is like this, it is going to be elongated under the influence of auxin. Apart from that, apical dominance. What do you mean by that? Basically, it does not allow the formation of lateral branches. Apical means apex is going to be dominating over here. There is so much amount of auxin over here. It does not allow the lateral growth to happen. Suppose this question comes to you. That is, a farmer wants to have a bushier plant. A bushier plant means so many branches. At that particular point of time, what should he do? He should be cutting up the tip, because the tip, what does he have? Auxin, and auxin is responsible for apical dominance. Got it? Apart from that, root initiation and also fruit development is done with the help of auxin itself. Okay, so cell elongation and apical dominance, please do not forget it. That is the most important part of all. Now gibberellin, my dear children, it is responsible for stem elongation. Stem elongation, apart from that, seed to germinate, gibberellin, germination. Okay, for that you need, and also the fruit setting and all, you need gibberellin. So at least this and this you need to remember for sure. Cytokinin, cyto means cell, kinin means division. So it is the one that is going to stimulate cell division, and also one very important thing, it is going to delay aging. That is what you call as senescence. So delay senescence and promote what? Lateral shoot growth. It allows the lateral shoot growth to happen. That is the role of cytokinin. 3 of them are growth promoting. Now 2 of them are growth inhibiting. One is ethylene. As I said, it is responsible for fruit ripening, and it does the opposite of what cytokinin does. Cytokinin delays senescence, but over here it is going to lead to senescence, my dear children. Okay, and it is also going to lead to leaf abscission. Leaves are going to be falling down as well. Now abscisic acid induces dormancy, so it is going to do the opposite function of gibberellin. Abscisic, you can remember like that. Both of them are going to do opposite function. Abscisic acid, which is there, it is going to induce dormancy. It does not allow the seed to germinate. Stay here, because no water is there. You have to be staying here. It's a stress hormone, right? It closes the stomata under water stress and inhibits the growth. That is why you call them as growth inhibitor. Okay. Now, write the characteristics of PGR again. So you have to say they are organic substances produced in the case of plants. They regulate growth, development as well as response, act in very low concentration, can be growth promoters or can be growth inhibitors also. Fine. Now define the term. We have done it. Differentiation, dedifferentiation and redifferentiation. I told you, process where meristematic cells, as in the cells which keeps continuously dividing, they become specialized or they become mature, is known as differentiation. Ulta of it, dedifferentiation. Ulta of it, redifferentiation. Okay. So mention, please know the examples of it. That is also important. Mention the source from which the following growth hormones were first isolated. Auxin, you got it from oat coleoptile tips. You have to remember this, oat coleoptile tips. Okay, kinetin you discovered from herring sperm DNA extract. There was a fish, from that you got herring sperm DNA extract, from that you got the kinetin. Gibberellin isolated from Gibberella fujikoroi, that is a fungus. Gibberella fujikoroi. So auxin from oat coleoptile tips, kinetin from herring sperm DNA extract, gibberellin from Gibberella fujikoroi fungus. Done. Now, both growth and differentiation in higher plants are open. What do you mean by open? That means it is continuously happening. Growth also has to be continuous, because cells continuously divide, my dear children. And why differentiation is open? Because, my dear children, these cells definitely are going to do some function at the later end. These cells which are dividing, they have to do some function, which is why we say they are getting differentiated. I told you, right? Everything will be assigned a function. So that is the reason. Okay, now describe arithmetic growth. Growth at the constant rate producing a linear curve. So arithmetic growth means, my dear children, it is going to have something like this. It is going to grow. For example, root growth, you might have seen it is continuously growing, right? So root elongation is an example of arithmetic growth. Now geometric growth. Growth occurs exponentially, forming a sigmoid curve. So over here, yes, growth happens, my dear children, but later it is going to reach a stationary phase. So geometric growth occurs exponentially, forming a sigmoid curve, that is cell division in meristem. So cell division happens, but up to only a certain limit. Beyond that, it is not going to happen. Okay, now absolute growth rate. Measurement and the comparison of total growth per unit time is known as absolute growth rate. So my dear children, here, initially the growth rate which was there, it was 5 centimeter square, then it was 10 centimeter per square. So what do you say is the absolute growth rate? So you have to compare between both of them. So 10 minus 5 is going to become your absolute growth rate, because that is what is said, measurement and comparison of the total growth per unit time, total growth per unit time. So how do we know what is the total growth? We have to do this minus this. Initially it was like this, then it was like this. So 10 minus 5, it will be what? 5 centimeter square. That is going to be what? The growth rate out there. Apart from that, timing will also be mentioned. Now per unit time, okay, you have to mention that. Now relative growth rate which is there, the growth of the given system per unit time expressed on a common basis. So how do you express that? Listen to me. Over here, you have got the absolute growth. There is a formula that you need to remember. Absolute growth divided by initial growth into 100. So absolute growth over here was what? 5. Initial growth over here was what? 5 only. Initially it was 5 only. Into 100, it is going to be what? It is going to be what? So 5 basically over here. Yes, 20. So it is going to be 400. Yes. It is going, just a second. No, not 400, 100, sorry. Okay, it is going to be 100 over here. Whereas when you talk about this, we have to compare, okay? We have to compare this also. So absolute growth over here, let's compare. So it was 50, 50 initially. It was 55. Over here, absolute growth is 5 only. Now we talk about initial growth. Initial growth was 50. So 5 divided by 50 into 100. Here 0, 0 is going to get cancelled. You just get 5 into 2, that is 10. So here it is 10, here it is 100. There is a comparison that is happening up over here also. Okay, so that is what? Relative growth rate. The growth of the given system per unit time expressed on a common basis. Okay, fine. Now, chemical coordination and integration. List the functions of the thyroid hormone. What is the function of thyroid hormone? It is a very important hormone for all your metabolic activities. As in making of things and breakdown of things also. So these hormones also support the process of red blood formation. RBC formation is also supported by thyroid hormones, my dear children. Thyroid hormones control the metabolism of carbohydrates, proteins and fats. As I told you, the breakdown of carbohydrates, proteins and fats, and also the making of carbohydrates, proteins and fats. Now maintenance of water and electrolyte balance is also influenced by thyroid hormones. The maintenance of balance of water and electrolytes, that is also maintained, and also, my dear children, it is responsible for production of thyrocalcitonin, thyroid gland which is there. It is responsible for production of thyrocalcitonin, which is another hormones. Apart from thyroxine and triiodothyronine, we have thyrocalcitonin, which is responsible for regulating the blood calcium level. Okay, so thyroid hormones, basal metabolic rate, the first thing, okay, they are helping in what? RBC formation and basically metabolism of carbohydrates, proteins and fats. And apart from that, it is the water and the electrolyte content is also balanced by this particular hormone. Apart from that, there is another hormone, that is thyrocalcitonin, which is responsible for regulating the blood calcium levels as well. Got it? Yes. Okay. Now, list the hormones secreted by the following: hypothalamus, GI tract, thymus, pituitary and adrenal. Listen, hypothalamus is the bap of all, as in the grandfather of all. Whatever hypothalamus says is followed by pituitary, and pituitary is going to release some other hormones which will be having effect on all the other glands. Okay, so basically hypothalamus is at the top. So hypothalamus can be releasing TRH, that is thyrotropin releasing hormone. When the thyrotropin releasing hormone basically is there, it goes and tells the pituitary to to release thyroid stimulating hormone. To release thyroid stimulating hormone. So basically, TRH over here goes and tells the pituitary, and now the pituitary is going to release TSH, that is thyroid stimulating hormone, that is going to act on the thyroid gland, and thyroid gland will be releasing some hormones out there, thyroid hormones out there. That is how it goes. Now you have GnRH, gonadotropin releasing hormone. Again, GnRH is going to act on the pituitary, and the pituitary is now going to release FSH and LH. Now GHRH, growth hormone releasing hormone. Growth hormone releasing hormone goes act on the pituitary. Now growth hormone is directly being released. Now CRH, corticotropin releasing hormone, it goes and acts on the pituitary and it releases ACTH. You get it? So basically all of this hormone can be releasing over here. So over here I have written releasing, but it can be inhibiting hormone. So in that case, it is going to inhibit the secretion of TSH or LH and FSH and things like that. Depending on whatever it's needed, hypothalamus is going to release that. Okay, now in a gastrointestinal tract, you have multiple hormones. For example, gastrin, it acts on the gastric glands and it stimulates the secretion of what? HCl. That is the role of gastrin. Secretin acts on the exocrine part of pancreas. Pancreas release enzymes, right? So exocrine part of pancreas, secretin is going to be releasing, stimulates the secretion of water and bicarbonate ions. So it says that, hey, you have to release water as well as bicarbonate ions. Now, cholecystokinin acts on both pancreas and also gallbladder. On pancreas, it acts for the secretion of what? Pancreatic enzymes. On gallbladder, it acts for the secretion of bile juice. Now, gastric inhibitory peptide, this is going to inhibit gastric secretion and motility. So basically, gastric inhibitory peptide will have a opposite function of what gastrin does. Cool. Fine. Now, what do you have? Thymus. Thymus, my dear students, it's over here. It's behind our sternum, behind our rib bone over here. So there's a middle bone or chest bone which you call as, okay, it is also known as sternum. Just behind that you have a small little thing that is known as thymus. This gland secretes the peptide hormone that is known as thymosins. Okay, now this thymosins which are there, they take part in differentiation of T lymphocytes. So T lymphocytes which are there, a type of immune cells, they are going to mature up over here and they are going to be differentiated over here. Maturation, differentiation is the same thing, okay. That happens in the thymus. Only T lymphocytes maturation happens over here. Now pituitary, anterior pituitary gland is there, posterior pituitary gland is there. As I said, whatever the hypothalamus says it is being followed by the pituitary. So it can release growth hormone. This is a direct hormone which is made in the pituitary and basically is being released also. Prolactin, similarly, thyroid stimulating hormone acts on the thyroid gland then. Adrenocorticotropin hormone acts on the adrenal gland. Luteinizing hormone, its own hormone. Follicle stimulating hormone, its hormone. Melanocyte stimulating hormone. So these are the things that are being released by the anterior pituitary gland. Now posterior pituitary basically is responsible for storage of things. So hypothalamus secretes something and stores in the posterior pituitary, and those hormones are oxytocin and vasopressin. Okay, these two actually are hypothalamic hormones, but they are stored in the posterior pituitary gland. Fine. Now, adrenal glands are above your kidneys. They are paired structures out there, and they release adrenaline, that is epinephrine, or noradrenaline, that is norepinephrine. They are responsible for fight or either flight hormone. Okay, cortisol helps in metabolism and stress response, and aldosterone regulates the sodium and the potassium balance. Aldosterone, sodium and potassium. Cortisol, which is there, metabolism and also stress response. Okay. These are the basic things that you need to remember. Now, write one physiological function of LH, luteinizing hormone. Luteinizing hormone in the case of females, my dear student, it is going to make sure that ovulation happens. Okay, ovulation happens, and also in the case of males, it is going to be responsible for testosterone production because they act on certain Leydig cells. Leydig cells are responsible for testosterone. So basically, testosterone production, that is a male sex hormone production. Now, growth hormone is responsible for a whole body growth. If at all it's not there, you have dwarfism. If at all it's too much, you will have gigantism. Now, oxytocin, it induces labor contractions in the uterus to contract and relax, you need oxytocin. Again, remember, hypothalamic hormone, but stored in the posterior pituitary. Vasopressin, it is anti-diuretic hormone. Now, if at all you want to control the water retention in your body, my dear children, through urine, obviously, there is vasopressin which is going to act accordingly, okay, so that the water can be saved. Sometimes water can be lost also. Now TSS, thyroid stimulating hormone, regulate the thyroid hormone secretion, that is thyroxine, triiodothyronine, thyrocalcitonin, etc. Now a sample of urine was diagnosed to contain a high content of glucose and ketone bodies. Okay, glucose is there, that means carb is there. Okay, based on this observation, answer the following. Which endocrine gland and hormone is related to this condition? Pancreas. Why pancreas? Because there is a hormone, that is insulin, which is actually responsible for conversion of glucose to glycogen. When this happens, suppose in the blood there is too much amount of glucose that is converted to glycogen, that means you are lowering down the blood glucose level. You get it? So that has been done by the insulin. Suppose insulin is not it's not being produced properly because your pancreas are not functioning well. What is going to happen? The level of glucose is just going to be there, and it is going to be excreted out through the urine also. You get it? So that is the thing. Now name the cells on which this hormone acts. These hormones are going to act on liver cells, because in the liver the glucose is stored in the form of glycogen. In the muscle cells also, okay, and also in the adipose tissue, that is the fat cells, because obviously in all these places, my dear children, there is a conversion that is happening up there. Yeah, so please remember that. Now what is the condition called? It is known as diabetes mellitus. And how can it be treated? By providing insulin therapy. So if at all your body is not producing enough amount of insulin, you can get injections of it, or you have other multiple techniques out there. Okay, yes. Now, these are the other questions that you have in the 5-marker category. Schematically represent the action of estrogen. Name the endocrine hormone. Okay, name the endocrine gland in which all these hormones are synthesized. What are gonadotropins? Gonadotropins basically are what do you call? What do you call this thing? Estrogen, progesterone, all of these things, my dear children, they are gonadotropins. Okay. And write a note on pineal gland. Catecholamines are hormones of flight and fight. Please understand it's norepinephrine and epinephrine. And enumerate the role of ovarian hormones as an estrogen and progesterone. And glucagon and insulin are antagonistic in their action. They are opposite in their action. Insulin converts glucose to glycogen, and glucagon converts glycogen back to glucose whenever we need energy. Suppose I am fasting, I need energy. So whatever stored glycogen is there in my body, that is going to break down into glucose. That is what's the function of glucagon. So please know about that. Okay. Yes, just a second. Okay. Over here, gonadotropins are not estrogen, progesterone. Gonadotropins basically are FSH and LH. Okay, FSH and LH. Okay, just to make you clear that. Now, the next chapter that we have is locomotion and movement. Over here, there is a 7-mark chapter. It was like that. And the 5-marker probable question that usually used to come is, what is skeletal system? Right, it's 2 divisions. So, skeletal system provides support, movement, protection, and acts as a storage site for minerals like calcium and phosphorus. It consists of bones and cartilages and is divided into 2 things. One is your axial skeleton, which is going to make the most part of your thing. That is your skull, your vertebral column, your ribs, as well as your sternum. There are 80 bones out there. Please know about them, more about them. You have to write more. Okay, then only you will get the 5 marks. And then here this appendicular skeleton, 126 bones are over here. That includes your limbs, that is forelimb and hind limb, and also the girdles are there, pectoral as well as pelvic. Okay, write more and get more marks. Just an understanding of what should be definitely there. Okay. Now, endless the bones forming the forelimb. So, in the forelimb, my dear children, you have the humerus here, and then you have the radius and ulna. You would have the carpals, you would be having the metacarpals, and then you would be having the phalanges out there. So, please know how many numbers of bones are there. Carpals 8 bones, metacarpals 5 bones, phalanges, you have 14 bones. They are finger bones. Okay, so please know about all of them. What are wrist bones? It is carpals. What are palm bones? It is basically metacarpals. So, yeah. Name some skeletal system disorders. Osteoporosis. It basically happens when there is decreased bone density. Why? Because there is a calcium deficiency that is taking place in your body. Now, arthritis, inflammation of joints leading to what? Pain as well as stiffness. Arthritis is what? Inflammation of joints that leads to pain as well as stiffness. Then we have gout. Gout is a condition where in your joints, in between your joints, there is too much deposition of uric acid, and that leads to swelling as well as pain. Okay, rickets is definitely vitamin D deficiency leading to weak as well as deformed bones. So, all of them has a problem with your skeletal system. Now, the 3 types of joints and the location in the human body. 3 types of joints: fibrous, cartilaginous, and you have synovial joints. Fibrous is the one that cannot move, immovable joints. For example, over here, in between the skull, you have joints which joins the skull bones. Do you think you can move it? No, Baba, you cannot. So that is the fibrous joint that is there. No movement, bones are tightly joined by dense connective tissues are there. Skull sutures as well as teeth sockets are examples for that. Now, cartilaginous joints, which are slightly movable. So basically, these joints are present in between our vertebral column, which is why we are able to do like this. Okay, slight movements are still happening. Bones are connected by cartilage. Now, whenever there is cartilage, obviously there is going to be movement. So over here in a pinna also, you have cartilage. okay, allowing limited movement. Example, intervertebral disc and pubic symphysis. So that's the places where you have the cartilaginous points. Now you have synovial joints. They are freely movable joints out there. Over here you have, over here you have, get it? Over here you have. So highly movable joints filled with synovial fluid that reduces friction. That is why we are able to move to certain more extent. Ball and socket joint, that is in the shoulder. So there is a socket over here, and the humerus, the tip of it is going to act like a ball. So we have a ball and socket joint. Allows movement in all that direction. Hinge joint is there in between your knee and also the elbow. Allows movement only in one direction. Can you take it back? No, you cannot take it back, right? Now pivot joint is there between the atlas and the axis. That are two vertebrae over here, and allows rotation of the head. Yes, it does. And gliding joint, wrist and ankle, my dear children, allows sliding movements. And then there is saddle joint, which is in the thumb. It allows the movement in almost two direction. Okay, so yes, have that understanding. Yeah. Now these are the other questions that you have. Describe the structure of actin and myosin. Explain the steps involved in sliding mechanism, muscle contraction. Important. Describe the bones of the skull in the man. Difference between skeletal and cardiac muscles. Sliding filament theory. Sketch the diagrammatic view of human skull. All of it. Cool. Now, cell cycle and cell division, this chapter again you have a one-shot video that I have done. If at all you want, you can watch it. So, I have done this one-shot video for 3 chapters the previous year. One was animal kingdom, one was morphology of flowering plants, and one was cell cycle and cell division. If at all you want, you can go ahead and watch those videos if you need, okay. You can just put master stroke and then master stroke, and then you can just type it down, the name of the chapter, okay. It will be coming up. So 3 chapters I've done. These are the questions that you have to anyway do. Please take a screenshot of this. This is a 2 marker. These are the 3 marker questions. And these are the, okay, 2 and 3 marker questions usually come from these chapters. Okay. Now we have respiration in plants. These are the questions that you should be aware of. These are 5 marker questions. Krebs cycle, citric acid cycle, TCA cycle, all of them same thing only. Different names. Know about the cycle. Again, as I said, please take a screenshot of this. This also, biomolecules, these are the questions that you should be definitely practicing. Listen, few questions I have done in model question paper. By seeing this, I remembered it. Please go and watch the model question paper video if at all you think that makes sense, okay. In that, I have explained each and every question, because obviously that is not going to be too long. So this was obviously not like that. So yes. Now, body fluids and circulation. Yeah, this chapter also I have done in master stroke. You can go ahead and watch it. Yes. Breathing and exchange of gases, these are the questions. Photosynthesis in higher plants, these are the questions. These are the 5 marker questions. Plant kingdom, these are the questions, 5 markers. Excretory products and elimination, these are the 3 marker questions. Morphology of flowering plants, this, as I said, I have done in master stroke. You can go and watch it if at all you feel it's difficult for you. And neural control and coordination, 5 marker questions, these are the questions. Biological classification, 4 marker chapter. 3 marker questions which usually comes up are these. And anatomy of flowering plants, these are the questions that you can have. Structural organization in animals, these ones. And the living world, these questions. And I think that ends up the whole video. You will get the PDF. You can just ask me in the comment section. I can provide it to you. And my dear children, I do not know if at all how much help I could be at this particular point, but at least I could guide you in some ways. Yes, if there are some concepts which you cannot understand by yourself at all, please put down their names in the comment section. If I can, I'll definitely make a video on that as well. Okay. So yes, I hope you are doing great and wishing you all the best. You will do good. And do not worry about anything, okay? So, bye-bye, take care. And do not forget to hit the subscribe button if at all you think this video helped.